So let's get going. Our first speaker is Dr. Amelia Johnson, and Amelia is going to talk to us about some really terrific work that's going on with respect to cancer and specifically looking for a cure for cachexia. And I'm so looking forward to hearing you talk about this. It's really groundbreaking research and kind of research that saves lives. So welcome, Amelia.
Thank you. Great. So thank you all for coming. I've been really looking forward to giving the presentation today, more so because this is a project that we've had ticking along in the background since the end of 2007, but it's really only in the last 12 months or so that we've been able to actually speak about the work and present it publicly. And the main reason for this is because it's a project that's always had very much a commercial focus. So in the original early days, it was funded by what we called a cooperative research centre for biomarker translation. And this was basically bringing together government and industry funding for projects with a commercial focus. And in particular for us, it was about developing a therapeutic against disease. And so what I thought I'd do for today, rather than just jump into what is cachexia and what is it all about, I thought I'd take a slightly historical approach to the way that I tell the story, because it's certainly a project that didn't start anywhere near where it's become, the story now. And so in order to take you back through the evolution of our project, I first need to introduce the key player in the story, which is a small molecule or protein called FN14. So what is it? And why were we interested in studying it all those years ago? So we know that it's a protein that lives on the surface of cells. And so this is a representative of any cell within the body. And here's FN14 on its surface. We know that in normal and healthy tissue, it's either at very low levels or it's absent. Now, it has a number of very beneficial roles in the body. Some of those being, for example, in inflammation, in the wound healing process, and in the regeneration of tissue after injury. And so what happens when one of these processes needed to be switched on is that FN14 is made. So here is some more of it on the cell surface. And it actually works in partnership with another molecule that's called tweak. And I'm not going to talk a lot about tweak today because it's very much an FN14 focused study, but just to show you that this molecule called tweak binds to the FN14, effectively switching on those processes when they're required. When those jobs are done, the levels then go back down. And so where there's a role for a protein in normal health, there's quite often then association in disease. And this is certainly not any different for FN14. There's an extremely long list of diseases that it's been associated with. We're particularly interested in cancer, and so this is the one that we'll focus on today. And so from this information, FN14 is, we really thought has good potential as a target for disease therapy. And the reason, just to add another piece of information, so I don't need you to see all the detail in this slide, but I just wanted to show you that, so FN14 and its partner molecule tweak, they're members of this very large family of proteins. And the piece of information I wanted you to take away from this slide is that this family of proteins are all very much heavily associated with different disease states. So from a therapeutic development perspective, they're a good group of molecules to be following. And when we look at the current therapies that are either in development or currently being used in the clinic, you can see that there's quite a large number of them. So these are a really hot family of proteins in terms of their commercial potential. To take it one step further, this is one such example. This is TNF. So it was probably one of the earliest discovered and certainly the best described and the furthest in development in the clinic. So this is just I pulled a list of the best-selling biotech drugs for 2014. As you can see, so these are the 3 TNF therapies that are currently in the clinic, and they are 3 of the top 4 in that list. So from a commercial potential, you can see, I think the dollars speak for themselves. They're certainly a very successful therapy. So we certainly felt that targeting FN14 could certainly be up the right type of protein to focus on. We were interested in knowing more about FN14's role in tumours, and the reason, so we certainly know a lot more now about its role in tumours, but when we started the project, we knew that the levels of FN14, so if you remember, in a normal healthy tissue, they're either absent or at very low levels, whereas they've been shown to be increased in almost every solid tumour described. And so our hypothesis originally for the project was that FN14 could be a promising target for the treatment of cancer. And so in order to test this hypothesis, we had a 3-step plan. Step 1 was to generate a model in the laboratory. And so this is a mouse model of disease, and it would be a tumour model that had an FN14 dependent component. We were then going to generate a very specific therapy targeting FN14, and then we'd take that therapy into the tumour model and see if it had an effect on the tumours. And so we went about making the tumour model. So we basically took a cell line and we created it in a way that if we injected this into mice, it would form solid tumours. We then added extra FN14. And so as you can see here, this is looking at groups of mice over time, and we're looking at the size of the tumours over time. And you can see that all the different groups grow at quite a nice rate, relatively comparable. And these ones here in red are the ones that have the extra FN14. So we made a model that had, grew really nice tumours. What we were really surprised to find, and certainly extremely distracted by, was this observation that, so this is the same 3 groups of mice, 2 control groups, tumours without FN14, and then a tumour expressing FN14. So pretty much from about day 8 of tumour formation, every single mouse in the experiment would begin this onset of really severe weight loss, loss of strength, and onset of really quite severe illness symptoms. So much so that they needed to be euthanized. It was that bad. And so this was really surprising to us, and more so because it's something that had never been described for FN14 or even suggested by other groups. And so we were a bit surprised, excited by what we may have found, but certainly we spent a lot of time after these original findings really validating what we had, understanding what the disease state was that we were working with. And basically, the point was that it's causing this disease that we call cachexia. And so now I'll tell you a little bit about what cachexia actually is. And so it's a strange sounding word, and it's actually derived from Greek words kakos and hexis that mean bad condition. And I think that's a really perfect way to describe it. So the clinical definition, it's been described as a complex metabolic syndrome associated with an underlying illness and characterised by the loss of muscle mass with or without the loss of fat mass. Anorexia, inflammation, insulin resistance, and increased muscle protein breakdown are frequently associated with cachexia. And so because a picture speaks 1,000 words, I've chosen, this is an image of Steve Jobs, who many of you may be familiar with his face. He was the founder and CEO of Apple, who passed away in 2011 from pancreatic cancer. And this is an image of him in his earlier times, looking quite healthy, and then an image of him in his very late stage disease. And the picture speaks for itself. This is a really severe condition that cachexia is. And so it's associated with a number of different disease states, a lot of chronic diseases such as chronic heart failure, kidney disease. As I mentioned, we're particularly interested in cancer. So when we look at the cancer statistics, we know that up to 80% of cancer patients with advanced cancers will suffer from some degree of cachexia. And it's up to 80% because in the more aggressive types of cancers, you get very high percentages of patients with cachexia. In the less aggressive cancers, you have lower percentages. So we also know that an estimated 20 to 30% of cancer-related deaths are because cachexia is present. Most significantly, there's currently no FDA-approved drugs for use in the clinic against cancer cachexia. And so it's a huge unmet need, and it's a condition that really needs more therapies designed to overcome it. So, what would a successful anti-cachexia therapy look like? What would it mean for patients? So it would mean increased quality of life, increased strength, the ability to maintain body weight, the ability for a patient to continue to just perform their normal daily tasks. In addition, the ability to continue to receive anti-cancer therapies. So you can imagine, when a patient becomes so cachectic, they're so weak, they simply just can't continue their cancer therapy. So if we could, create a drug that would help that process, then they could continue to fight the cancer. So overall, improved prognosis, better survival rates. So we'd made this really interesting observation that FN14 was able to cause this cachexia. The next thing we needed to do, and because it was part of our original plan, we were generating therapies on the side against FN14. And what we chose to do to make one of these is we chose to make what's called a monoclonal antibody therapy. So now antibodies are molecules you may have heard of. So they're a very natural molecule to the body. These are what the immune system uses to fight infection and disease and to also protect self. And so what we can do is actually utilise that biology, but we can do it in a way that we're generating those in the laboratory. And the reason that we like monoclonal antibodies is because they're very, very specific for one target. So just to give you an idea what an antibody looks like, this is just a very basic structure. So this region here, all antibodies have, this is called a constant region. And then this region on the end that we call a variable region, this is the part that makes them very specific for one target. And so by having this really high specificity, what it means is that there's much less chance of off-target side effects. And so antibody therapies are quite common in the clinic now. There's currently 36 of them approved, and so they're certainly a good type of therapy to generate. When we set about to make these antibodies, having an antibody that recognised FN14 was what we were after, but just not any sort of antibody. We weren't particularly interested in antibodies that were able to bind to FN14, but not really do anything to its function. We weren't also particularly interested in generating antibodies that were able to bind to FN14 and switch on those functions. What we were looking for was antibodies that had the capacity to bind to FN14 and effectively switch off whatever's happening. And so we were successful in making a number of these. And basically how we did this is we immunised mice with some FN14 to raise an immune response against the protein. We then take those spleen cells, so this is where the antibody producing cells are, and we fuse that to a cell that can grow indefinitely in culture. And so basically what we make is little factories that can pump out an antibody. So we made a number of them. And so we then took our antibody into our tumour model, but instead of looking at the effect on tumours, what we wanted to see is whether it would block this cachexia. So again, what we're looking at in this particular graph, this is the average body mass for each group of mice. Each of these lines represents a group. And you can see in the dark blue, this is a control tumour. So there's a mice bearing a tumour without FN14 on the surface. You can see they maintain quite a nice healthy weight. And then if we inject our antibody treatment into a group of mice carrying those control tumours, there's very little difference in the weight over time. And this basically showed us that there's no, at least visual, obvious effects of any toxicities. So this group in red here, this is our FN14 expressing tumours, mice suffering from cachexia. And I remember when we first ran this experiment, we talked about, given the severity of the disease and the short time frame, what would we like to see that we would call a positive outcome? And what we discussed was, look, even if we saw a couple of days extra survival, this would be a bonus. We were shocked to find that if we administered an antibody twice a week and kept that antibody level up in the mice, they were basically able to survive relatively healthy, no signs of illness for the length of the experiment. And this is even given the fact they had quite large tumours still. So this was a really striking finding. We were really excited about it. And so it also asked that what we needed to do. So this model we'd made ourselves in the lab was relatively artificial in nature. What we needed to do was then take our findings, take our antibody and put it in a model that's very, a more generic type of model, something that other cachexia researchers use. And so we did that. We established a model that's very commonly used called the colon 26 model. And we were in fact able to show the same results. So here again, we're looking at body mass over time of each of the groups. These are mice bearing the colon 26 cachexic tumours. And then in blue, when we add our antibody to a group of those mice, we can completely block the disease state. So this was a really exciting finding. If we look at that in terms of survival over time, you can see again here the cachectic mice not surviving beyond this point here, whereas those that have been treated with the antibody are able to survive a lot longer. And I was always a little disappointed with this particular graph because there's a couple of animals here that were dropped out of the study. Unfortunately, they were dropped out for ethical reasons and were actually not suffering from cachexia. So if we took those ones out of the graph, it would have actually been 100% survival. So just in summary of what our findings have shown, we've shown that when FN14 is expressed on the surface of a tumour, through this process that we really don't understand very well yet, it's able to induce this cachectic state. We're able to generate antibodies that were very specific for FN14 that could shut down this process, allowing the mice to retain their body mass, retain their muscle mass, and basically revert from a cachectic state. And so where to next? So in an ideal world, it'd be nice to think, let's take our antibody and let's put it into humans now. And this is certainly our plan, but there's really a lot more steps that are required between now and actually starting human trials. And this is actually probably a really good time to introduce a very important collaboration we have with Dr. Andrew Scott and his team at the Olivia Newton-John Cancer Research Institute, who are helping us drive the work forward from here. And so we have, a number of different questions that we need to address. So we know that FN14 triggers the cachexia. We know what the downstream symptoms are. What we don't know is everything that happens in between. So this is a really important thing for us to really understand what molecules are involved and how it all works. It's important because this will help us translate what we've got in our mouse models into the human condition. And that'll also assist us in understanding, which patients that are suffering from cachexia are going to be the most likely to be responsive to an anti-FN14 therapy. And so then in terms of development of our antibody for trials, basically what we have currently is a mouse antibody. And so this is not appropriate to put into humans. So what we need to do is basically recreate that antibody in what we'd call a human format, or a humanised format. Once we've done that, we need to then go and show equivalence to our mouse antibody, make large amounts of this humanised antibody to then go ahead with all of the formal preclinical studies, such as toxicology studies and what we call pharmacokinetic studies, because we really need to understand how that antibody will behave. That's the point where we're then able to enter phase 1 and 2 trials. And so what we're looking at here is probably a time frame of 2 to 3 years. And just before I head to my acknowledgement slide, I just thought I'd share with you, if I can find it, this little video.
I see cachexia, and you'd imagine that I would see it rob people of the chance to fight, to function, to be. That I would see the strength needed for the simplest tasks disappear, replaced with words like, I just can't, I just can't. That I would see faces change, cheeks becoming hollow, skin sagging from bones. You'd imagine that I'd see faces grow unrecognisable, slowly but surely, even to themselves. I see the struggle. I do see people disappear. But I also see the undeniable want to keep living. I see so much more than what you'd imagine. I see smiles, love, humour, the human spirit, hope. There is an antibody being developed that, if successful, will give people a better chance to fight, to function, to be. That day is coming, and I'll be here, waiting, when it does.
Excellent. So just to give a bit of an overview of all of the people that have been involved, just quickly some of the key players. So Nick Hugenrath, who's here today with us, he and I lead the project. John Silk, who originally initiated the project and led that in the first few years. Laura is one of our researchers who's been with us almost the entire project. The rest of our current and past team here at La Trobe. Andrew Scott and his group at the Olivia Newton-John Cancer Research Institute, and all of our other collaborators around the world. And very importantly, our funding bodies. Obviously, without the funding, there's not research that happens. Notably, more recently, a Victorian Cancer Agency large translational grant that we're working from. And thank you all for coming.
Thank you for a great talk and for keeping to time.
Oh, did I?
Yes.
Great.
So if anyone in the audience has some questions for Amelia or discussion points they'd like to raise. Yep, Andy.
That was fascinating. I think if I understood correctly, you showed that antibody treatment could prevent cachexia. Is that correct?
Yeah.
Is there any indication that the antibody treatment could rescue a mouse who's suffering cachexia and bring them back to a normal body weight?
Sure. So I only showed a few pieces of data today, and those studies were treatments on the day when we expected to see the weight loss begin.
Yep.
We have done studies where we've treated, so obviously, you know, that time frame between onset from a perfectly healthy looking mouse to the end point is quite normally about 3 to 4 days. But we have treated about probably 2 days into that and shown that we're able to rescue a number of them. Yeah.
Other points? Questions? Yeah.
Is the anti-gen, or the thing that's on cell, is that an independent parameter for using prognostication of various tumours?
Sure. It's certainly, it's been studied on tumours as being present to different degrees. Whether it's able to be used as a prognostic, at least for this particular use, we're yet to look down that path, but it's certainly something that should be explored.
Some types of cancer, particularly prone to cancer,
Sure.
and you would think that possibly they may have all this antigen on their tumour surfaces.
Yeah. So it's something that we'd like to look at. It's been a feature that's been difficult to find. So there's a lot of databases out there of patient samples, and so we know the levels of FN14 and what they look like on different tumours. What those databases are generally lacking is information about whether a patient suffered from cachexia or not. So that's where we've, we really need to form new information so we can actually make those correlations. But certainly from an indirect point of view, if you look at the types of cancers where FN14 is expressed and more highly, and compare that to the types of cancers you see higher cachexia incidences, they're generally quite overlapping. So the more aggressive ones have FN14, the more aggressive ones have more cachexia present.
I've got a question for you about funding. So I assume you have some grants under review at the moment.
We do,
Yes.
yes, yeah.
And what's the plans? If they all came through, what would you be looking at? What grants have you got going?
We've got an NHMRC development grant that we're looking forward to hearing on. And we're also, you know, we've got grants that we're looking at. We're also in a process of fundraising for the commercial development.
Yeah. Well, it's a truly magnificent finding, and there's obviously an incredible amount of people that have been behind this work. We look forward to hearing what happens in the future. I just hope you win that funding so it can happen a little more quickly.
Thank you.
Thank you so much for your talk.
Thanks.